Microscopy. Magnification is the number of times bigger the image is compared to the specimen. Resolution is the minimum distance between 2 points in the specimen where they can still be distinguished as separate. The smaller the distance, the better the resolution. Preparation techniques. Fixing. Preserving structures by making cross links in tissue, using chemicals like glutaraldehyde. Dehydration. Drying out tissue using solvents like acetone, nail polish remover. Embedding. Injecting in a resin, E.G araldite, into the specimen to make it hard. Sectioning. Slicing the specimen very thinly using an ultramicrotome, a special guillotine. Staining for electron microscopes is done by coating the specimen in heavy metals, E.G osmium tetroxide, so the electrons will be reflected and not transmitted. Differential staining for a light microscope uses a combination of stains like iodine or nile blue to stain some cells or tissues to make it easier to see particular structures inside cells or particular tissues, as the densities of different organelles mean different amounts of stain will be absorbed. Types of microscopes. Light microscopes. Light is used to form an image, limiting the resolution to half the wavelength of light. Maximum resolution is 200 nanometers, so they can only be used to view eukaryotic cells. Maximum magnification is about x 1500. Denser parts of the cell will absorb more iodine stain, providing more image contrast. Electron microscopes have a better resolution as electrons have a shorter wavelength than light. They also require a vacuum, otherwise electrons will bounce off other molecules. Transmission electron microscopes, TEMs, use electromagnets to focus a beam of electrons to be transmitted through the specimen. Denser parts of the cell absorb more electrons, making them darker and providing more contrast. Higher resolution allows their internal structure to be seen. Specimen must be thin, dead, and dehydrated. Image has no color. Lengthy treatment means artifacts may be introduced. Scanning electron microscopes. Electrons are reflected off the surface of the specimen, which is coated in heavy metals. The bounced-off beams are detected, forming an image. A 3D image is produced of the specimen surface. Lower resolution and magnification than TEMs. Laser scanning confocal microscopes. A high-light intensity laser beam is used. Specimen are treated with a fluorescent dye. Fluorescence is the absorption and re-radiation of light. The specimen, such as a thick section of tissue or small living organism, is scanned with a laser beam which is reflected by the dye. Multiple depths of the specimen are scanned to build an image. High resolution as laser beams are focused at specific depth but lower than TEMs. Can be used on thick or 3D specimens, allowing external 3D structure to be observed. Slow process, a long time to produce an image. Laser can cause photo damage to cells. Calculations. Image size equals actual size of object divided by magnification.